So Jalen London is going to go first, then Galen Jones, and then Kara Kuprantet. Say your last name, please.
You got that right, Kuprantet.
Oh my gosh. Okay, thank you. All right. And so let me read the bio, and then you can pull up your presentation if you would like, Jalen, since you're first, as I'm reading your bio, okay? All right, so Jalen London is an honors student studying nursing with a minor in Spanish, wow, at the University of North Carolina at Charlotte. He previously completed an online National Science Foundation funded research experience for undergrads program, which with the lab that he's currently working on. And he is a member of the Martin Scholars Program, a competitive merit scholars program. And you are here to present about silver nanoparticles. Wow. Okay, so go ahead and pull up your presentation, Jalen, if you don't mind, and take it away. And thanks again for everybody joining us. Please stay muted. We will ask questions at the end of Jalen's presentation. Thank you.
Okay. Yeah, so my project mostly has to do with silver nanoclusters as antibacterials. This is a project I worked on last summer within my lab, and it was very interesting, and I've been like kind of building off of it. Nowadays I kind of like looked at a different approach and different stuff, but I think it's just very relevant to kind of share this idea in the biomedical field. Okay, so the background, the reason why we're looking into this research is because bacteria right now, antibiotics aren't the best. A lot of times they attack the beneficial bacteria in our systems, and also they allow for new mutations, and they quickly mutate. So we're looking for a more like biomedical, like healthy approach to this research. And my lab specifically looks into nucleic acid nanoparticles. And the way we do that is we change the shape of these nanoparticles so that we can introduce other therapeutics, which is what I did. We looked at lectins and quantum dots. And the way we do this too is just like manipulating the shape, whether it be with the DNA or the RNA, to create shapes such as nanorings, nanocubes, duplexes, or fibers. Okay, so my methodology for what I did specifically in my project is that we were looking to use silver nanoclusters, which is just small silver nanoparticles within a small like little cluster shape. And we used a DNA ring, which is just like a DNA ring around it. And then what we're doing is we're trying to see the binding ratios to see specifically like how many silver nanoclusters are binding to each bacteria. So we did this in 3 different steps. We looked at the best binding ratio of quantum dots to biotinylated duplexes. Then after that, we looked at the binding efficacy of strep-having encoded quantum dots to biotinylated silver nanoclusters. And last, we looked at the ratio of lectins to these quantum dots. And I'm going to go into a whole explanation of specifically what each thing is, but that's just the methodology, how we even started this whole project. Okay, and for verification throughout the entire project, we use gel electrophoresis. So we're just running electrical current through a gel with these different like components in it, and we're testing the like how far is it running, which would tell us the whole atomic weight, so we can see if it's actually binding or not. If it's binding, it's going to have a higher atomic weight, so we can see that it's higher than the other, just the control. So that's how we're mostly verifying if the ratio and the binding is occurring. Okay, and we also have to use Comosi blue for the lectins, because it's a protein and lectins cannot be verified just using the ultraviolet light. And then for the other components such as the quantum dots, we just ran it on a native page and used ultraviolet light to have like a visual representation of the quantum dots, which you can kind of see from the photo here. This is quantum dots in the silver nanoclusters and the stain, and they're pretty obvious to see exactly like the binding. It's not a great picture of the binding itself, but just a visualization of like what they look like using ultraviolet light. Okay, and it's just the creation of the silver nanoclusters to go into it. So it's just using DNA, which is already in a duplex structure or a ring structure or hairpin structure, I'm sorry. and we add ammonium acetate buffer, then silver nitrate, and reduce it with sodium borohydride, and then we visualize on the gel that I was referring to. So to go into the project itself and exactly what we're using, silver nanoclusters. So silver has already been used as a means of purification in food packaging, air, water, and treating textiles. It's an effective antibacterial agent. And but by us using a cytosine-rich section of the hairpin structure, we can attach the silver nanoclusters to this hairpin. So if you can see the loop region and those little yellow dots, that would represent the silver nanoclusters within the cytosine-rich hairpin, and the rest is just holding it together. So yeah. A quantum dot's just acting as a scaffold for all of these things together. So a quantum dot's just a nanoscale cluster of atoms composing of a shell in a core and shell, and we're using it as a scaffold for the hairpin and silver nanoclusters. It does use some of the quantum dots and other projects use like cadmium and other metals, which people will question if they affect the body in a harmful way, but we're using it at such a low ratio that it would have no effects, or we have already seen it have no effects on the body itself, and it doesn't affect bacterial growth, which is a good thing because we don't want it to affect the beneficial bacteria in our body. And the lectins are just proteins that bind to specific sugars. So this is going to allow us to bind to specific sugars on bacteria that we want to like attack, instead of just attacking all bacteria in our microbiome. This is very important just because we want to promote higher selective antibacterial efficacy, which is something that right now antibiotics aren't really doing. They're kind of attacking a general amount of bacteria in our body to kind of save us from these type of infections, but it's also hurting us in our microbiome too, because we won't be able to defend ourselves from other infections in the future until these microbiomes can grow back together. So what we're doing here is that we're using lectins that bind on the specific sugars on bacteria to promote a high selective antibacterial efficacy. Okay, and what is holding this together? The hairpin structure, the silver nanocluster hairpin structure, and the rest of the quantum dot would be the avidin biotin complex. So this is just a very strong complex between a non-covalent interaction between avidin and biotin. It's unaffected by temperature, altering pH, organic solvents, and other denaturing agents. This is very important because we do not want these structures to break apart in the body. And avidin has no carbohydrate, and it has a lower solubility than avidin. So we switched from using avidin throughout the project. We switched from using avidin to stirpividin, and it's especially good for lectin binding as well. So yeah. So these is the projects we ran and the experiments we ran. So first, we just wanted to see like at what ratio or at what concentration would these quantum dots be able to be visualized using ultraviolet light and not be too strong. So it's just a baseline experiment. So we used around 3 microliters at the quantum dot just to visualize, just to see exactly like if it's not too strong on the light. So we're not actually like overfluorescing the machines. And then the next test we used is the strep-having coated quantum dots in the biotinylated duplexes. So obviously in the future we use hairpins, but just to like in the beginning we used biotinylated duplexes. And using the experiment we ran above, we used it at different ratios to see at what ratio are these tending to bind the best. So they binded the best usually at the 1:12 ratio, which we can see as it ran the furthest around the 1:25 ratio to 12.5 ratio. It ran the furthest, which just means that it's binding the best because it has a higher atomic weight and it's running more. So, and then our next test was just an experiment to see if our temperature, if our procedure in any way is affecting the results. So we just did different tests at like room temperature for 30 minutes, 95 degrees Celsius for 2 minutes, room temperature, and then the room temperature for 3 minutes. Then we kept it in a colder or warmer environment, 40 degrees Celsius for 30 minutes. And then, 37 degrees Celsius for 30 minutes. And then we just wanted to see if it had really any true like big effects on the procedure, or if the procedure itself is affecting the results we're getting. And we noticed that like it didn't have a big effect on it, which is a good thing too, because it's kind of showing the high binding affinity, meaning that you're not gonna have too much problems if you do like take this from a lab into a more clinical field too. So this is just a very interesting experiment. And our next experiment was seeing the binding efficacy of quantum dots to silver nanocluster hairpins. So this is when we're actually seeing like at what ratio are these silver nanoclusters binding to the quantum dots, and the quantum dots being the scaffold for all of this together. And what we noticed is around the 1:12 ratio. And if I can go back for 1 second, as you can see from here, that streptavidin and biotin bind, or this is already known, they're bind at a 4:1 ratio. So when we go in the future and we're looking at this, we can tell that the quantum dots and the streptavidin are binding at like around the 1:3 ratio, truly, just because of the biotin and the streptavidin is kind of like masking that a little bit. And then lectins is just obviously the protein that is binding to the sugars on the bacteria, which is giving it the specific binding. We didn't have amazing results on this yet, just because when we try to use Kamasi blue, it kind of masks the fluorescence of the quantum dots, so we couldn't see exactly when these proteins, Kamasi blue being a protein dye, we couldn't see exactly when these proteins are binding to the quantum dots. It's just because the Kamasi blue was masking the fluorescence. So we weren't exactly sure on that, and we kind of went into further testing with that. But we did see that they were binding at some point though, just because we used a ultraviolet light machine that allowed us to see that the quantum dots and the lectins are binding. Because if you look at the quantum dot control, it's not moving at all, just because quantum dots have no charge on the molecule. But the lectins, when they are binding to the quantum dots, like the quantum dots are moving down. So we are seeing the binding, we are seeing the current running through it. So in the future. So in conclusion, we're looking to move this from just binding ratios to actually like testing the accuracy on bacteria itself, and then furthermore on cancer cells, and then cancer cells, and we're testing on animals to see if they can affect infections on animals, and then eventually people. So that's where like the journey of this research would go, and it can help out with diseases such as pneumonia, tuberculosis, syphilis, and it can make sure that our immune system is not compromised at the same point. So it's just a great alternative to antibiotics as a whole. And then I was unable to continue working on this research, but my instructor, my person above me, was helping me. She continued working on this research. That's a part of her project. So in this project, she was experiment she was working on recently. She had tested to see if the lectins were binding onto the bacteria, just to see if it's actually truly binding, like if it's actually giving specific binding. And it actually worked out perfectly, as you can see on figure D. This is where the bacteria are, the E. coli. And then the figure E is where you can see the fluorescence of the bacteria, which is showing that the quantum dots and the lectins are binding onto the bacteria itself. And they're having very specific binding. So this is very promising for the future. And then in the future from this, like going on from this project or this experiment itself, we're going to looking at if it's actually going to kill the bacteria. So that's a very important part. This part didn't really test the silver nanoclusters, but just the binding of the quantum dot and the bacteria itself using lectins. And I just wanted to give a special thanks to Elizabeth Skelly. She was the person ahead of me, just looking over the entire project itself. And she was the one that had the initial plans for the project. And she's continued working on the project because I am now working on more cancer cell related research. Yeah. And just my overall lab that I work at. And thank you so much.
Okay, thank you, Jalen. Thank you for that presentation. Really appreciate it. Why don't we see if you have a question? Oh, we got some applause going on. Look at that. You can use your reactions. And if you have a question, you can raise your hand if you have a question. And in the meantime, Jalen, I have one question for you. So if you had to start over and do this differently, what would you do differently in your research?
If I had to start over and do this differently, I wouldn't say like we went too wrong anywhere, just because what we're doing right now is very baseline, just testing ratios. I wish we had to save a little bit of time with the protein dye. It's unfortunate with the Kamasi blue. It's just very masking. It's very hard to see if the lectins and the quantum dots are binding itself at what ratio. Kind of hard to tell. But other than that, like it's very baseline. And yeah, I mean, there's not much to change so far right now, but yeah, hopefully we can find other protein dye for lectins and stuff. That'd be pretty interesting. Hopefully we can do that soon.
Okay. Does anybody have any questions that's here in attendance? Oop, I think I saw somebody. Yes, go ahead, Abigail.
Do you think that this stuff might end up helping you with your cancer research? Because I think that would be really cool. This whole process was super cool. So if this could help you in your own stuff, that would also be awesome.
For sure. I wish it could. This is more have to do with like antibacterial stuff, and then what the cancer research is doing is more like cell work. So it kind of aligns a little bit, but like also like not too much. I wish it could too, but I mean, you never know. I think in the future it'd be very interesting to see like exactly if it's gonna work or not, and it could be applied. So I haven't applied it yet, but hopefully in the future I can. But thank you for the question. Great question.
All right, and Saria Co.
Thank you. I thought that was a really, really interesting presentation. And there was a 2020 paper that I've actually seen that looks at the health impacts of silver nanoparticles, specifically when it's reviewing in vitro and in vivo studies. And it was mentioned, of course, it is a fantastic antibacterial agent, and that is so important for us to be able to replace our current antibiotics. But one of the things it mainly covered was how in vivo tests have shown that silver can actually accumulate in organs, causing a lot of health effects. So I know right now, y'all aren't in vivo experiments, but you were saying that is a future direction you think this research could apply to. So how do you think y'all would maybe correct to see if this is causing issues or if the pros outweigh the cons when it comes to using this in vivo studies?
Yeah, that's a great question. I'm trying to think. That's a great question. Yeah, I did not know that like it affected the organism itself. I think what we were trying to do is like having the silver on the cotton dots and having like a scaffold for it to be on, so that it just doesn't like freely float in the blood system in our body. I think that'll help a lot with the flushing system. Yeah, I'm not exactly sure like if it would affect like it staying in the liver. Hopefully not, but yeah, I mean that's something that for the research. I appreciate that question, helping a lot.
Okay, and we have like a minute left, and let's see, actually, I'm a little concerned because Kara says she needs to leave at 1:50. So I'm wondering, Galen, could you go last because of that instead, and then have Kara go second, and then you can ask your question. Is that okay? Kara, is that okay with you? That way you're not feeling rushed. Okay,
That's fine.
sorry. I'm going to switch things up here. Sorry about that, guys. Okay, so Galen, did you have a question for Jalen then?
Yes, real quick, with the Kamasi blue being giving you issues, are y'all thinking about changing to a different type of protein, or are y'all still going to try to work out the kinks with that?
Yeah. So I guess like Kamasi blue is kind of like messing up the entire like with any protein per se. Like lectins are pretty like specific, and we're using WGA, which is the same type of specific lectin that we use for the E. coli. So I think we prefer to keep the same lectin itself, just because it's like E. coli is a very abundant bacteria they could easily get, so we can keep doing tests on it. So I think we're kind of looking at maybe even using a lower concentration of the Kamasi blue. I tried that before and actually helped a little bit. But yeah, I think we're more lenient with the changing the stain, the protein stain itself, than really just the protein, the lectin itself. So yeah.
Okay, thank you, Jalen. Great job. Congratulations. All right, so we're going to move then, Kara, to you. And if you want to go ahead and pull up your presentation as I'm reading your bio, that would be great. So this is Kara Kupratit. She's a second-year biology student at UNC Greensboro with concentration in human biology and chemistry minor. Wow. She is in both the Lloyd International Honors College and the Disciplinary College. And additionally, she is the founder and president of VAW. You'll have to tell me what that stands for. Global Health Alliance's Medical Chapter, and is on the Dean's List for 4 consecutive semesters. So welcome, Kara. And can you tell me what that VAW stands for? first?
Yes, VAW stands for Volunteer Around the World. So
Okay.
it's a student group organization that we go to low and middle income countries to help out in the clinics.
Okay. All right. Well, great. Welcome, and go right dive into it here.
All right, thank you. So my name is Kara Budding Credit, and today I will be presenting a past we cannot erase. Ancestral exposure to plastics will influence the health of future generations. And here's the order in this presentation that I will be going over. Every day, we interact with hundreds of plastics, such as plastic bottles, food packaging, automobile, canned food, hygiene products, and tableware. Within all of these products contain a chemical called bisphenol A, or BPA. And while BPA contamination began around 8 decades ago, its continuous use in mass-produced items has led to an environmental contamination around the world. And the widespread of BPA can cause a bioaccumulation in human and non-human organisms. BPA has also been classified by EPA as an endocrine disrupting chemical, or EDC, which means that they interfere with metabolism and disrupts hormones. And this is broadly concerning because the adverse health conditions associated with BPA exposure can have an effect in the fish and mammals, such as hormone disruption, thyroid cancer, lung cancer, infertility, and more. And these defects are the result of BPA's ability to modify the epigenome. An epigenome sits above the DNA, which controls what genes to be turned on or off in your cells to alter the gene expression. And our everyday environment can influence our epigenome, causing an epi mutations to change which genes can be turned on or off. Social status, diet, sleep, stress, or exposure to a certain chemical such as BPA can affect your epigenome, causing epi mutations. However, BPA-induced epi mutations are not contained within the individuals that were originally exposed. Instead, epi mutations can also occur in egg and sperm cells. That means all the health defects that I mentioned earlier can be passed down from one generation to the next, making them transgenerational epi mutations. I also want to go over some terminology before we get into this deeper in this presentation. So F0 is the first generation, F1 is the second generation, which is the children of the F0, F2 is the third generation, and F3 is the fourth generation. Perfluorooctane sulfonic acid, or PFOS, is a more modern chemical that also been classified by EPA as an endocrine disrupting chemical. And the exposure to PFOS can have the same or similar defects such as in BPA. And PFOS has oil and water resistant properties, which makes them really popular in manufacturing products such as food packaging, electronics, nonstick cookware, carpets, fabrics, raincoat, umbrella, which is a really great example in Greensboro today because it was raining. So we are being constantly exposed to PFOS. PFOS is also a member of per and polyfluoroalkyl substances, or PFAS, which has become environmentally relevant for the past 2 decades. And you may have heard its nickname as a forever chemicals because of its ability to maintain in the environment for decades after its initial use. Because of the constant presence of PFOS in the environment, water sources around the world has become contaminated with this chemical, and the extent of PFOS exposure resulted in CDC finding 98% of people, including newborns, have PFAS in their blood. And PFOS exposure is really relevant in North Carolina because it's considered one of the most contaminated states, and especially in Wilmington, more than 85% of children and adults have PFAS in their blood as a result of water contamination. A non-monotonic dose responses is a curve that looks like a U-shape or inverted U-shape, which is typically normal to see in the endocrine disrupting chemical, and we also will be seeing this in our results section. So now we have established that current and future generations have been exposed to BPA and they have a transgenerational epi mutations. Additionally, they are also exposed to another chemical, the newer chemical PFOS. And even though there's numerous studies on the effects of BPA and PFOS alone, there's no recorded studies that examine the influence of the ancestral BPA exposure on the sensitivity of the individuals experiencing the novel PFOS exposure. And so to understand the health risks posed to these health risks posed to humans because of these new chemical, we have to consider the realistic human exposure timeline and considering the ancestral BPA exposure. And we expect that individuals with ancestral BPA exposure and now experiencing to the PFOS exposure would produce an offspring with an elevated phenotypic malformations. And we use Medaka fish as our animal model because Medaka sex differentiation and processing epigenome is similar to humans. And here's the different stages of Medaka, but the one that we will be mainly focused on would be stage 40 and 41, which we call it fry or baby fish. We have two groups of Medaka. We have control group, which means that they were not exposed to BPA in the first generation or F0. And we have another group that were exposed to 10 micrograms per liter of BPA in the first generation. We let them grow and reproduce. And we have F4 generation, 3 males and 3 females, exposed each group to a relevant concentration of PFOS for 21 days during their reproductive period. And these relevant concentrations of 0.002 milligrams per liter and 0.02 milligrams per liter are found in our environment. We then let them grow and reproduce, and we collect the F5 embryos daily, and they were grown without any exposure to PFOS or BPA. We took a picture of them under the microscope, and we measured their phenotypes using MHJ and analyzed the data using t-test. Here is a picture of our correctly developed fries, so you don't have to concern about them. They are living their best lives. However, here's our developmental maldevelopment. So that we'll be focusing or presenting in this presentation. So there's uninflated swim bladder, which if they have that or don't have this posterior swim bladder, they cannot maintain buoyancy. There's yolk edema, which is excess fluid and the residual yolk. So if they have this, they have problem with circulation and movement. And finally, body length. Here's some of our interesting data that we found. So on day 5 of posterior swim bladder, as you can see here in our lowest concentration, the control group actually has more uninflated swim bladder than the BPA lineage. On day 5 of yolk edema, you can also see the same trend that in our lowest concentration, the control group has more yolk edema. However, when we increase the concentration, the BPA lineage, it displays more yolk edema. We can also see this trend on day 5 of body length, where in our lowest concentration, the control is shorter. And as we increase the concentration, the BPA lineage is shorter. On day 10 of posterior swim bladder, same thing. So in our lowest concentration, the control group has more uninflated swim bladder. And as we increase the concentration, the BPA lineage has more uninflated swim bladder. You can also see the same on day 10 of yolk edema and day 10 of the body length. So now you have seen the non-monotonic dose response in all of these 3 phenotypes, which is a U-shape or inverted U-shape, depending on how you look at it. You can also see that in our lowest concentration, the control group actually exhibit more or display more defects than the BPA lineage. However, when we are moving on to the higher concentration, the adverse effects are actually displayed in the BPA lineage. And that led us to a conclusion that the non-monotonic dose response observed in all of these phenotypes is usually a typical effect seen in most endocrine disrupting chemicals. And ancestral BPA lineages display a protective effect at low concentrations, possibly due to a prior exposure or adaptation from BPA, because you have to remember that BPA and PFOS are both endocrine disrupting chemical or EDC. So if the ancestral BPA lineage has been exposed to an endocrine disrupting chemical before, they might get used to it, adapt to it. So they kind of, when they have to expose to that PFOS, the EDC again, they're like, I'm fine, I've seen this before. So they kind of protect them. Or if you think about the vaccine as an analogy. And adverse effects occur at high concentrations of the PFOS. So as you can see in the higher end or the higher concentration, the BPA lineage actually has or display more defects. And maybe it's because they cannot deal with this high concentration. And this is concerning because this high concentration of 0.02 milligrams per liter is prevalent in the industrial areas, in agriculture, and in marginalized communities. So to link this to humans, it is harmful for women who is pregnant or will be pregnant because it's not only affecting them, but also their children and the future generations. Therefore, identifying this connection may lead to stricter regulations and further research to mitigate the impact of these chemicals on the environment and individuals living in these affected areas. For our future directions, we will be doing more analysis for longer exposure times, so day 15 and day 20. We'll also be doing more analysis for different phenotypes such as gray yolk, pericardial edema, and more. We'll also be going to be looking at gene expression analysis to examine the internal pathways, and the gene that we will be looking at will be acetylcholinesterase, thyroid hormone receptors, and thyroid stimulating hormone, and more. So in this presentation, we'll look at what's actually happening from the outside, so what we can see externally, but in the future, we will be looking at the internal, what we cannot see and what's actually happening inside of them. Here's my research references, and if you're interested, I can pull this up later. Thank you, Bandera Lab, for the continuous support, and thank you for listening to my presentation today, and I will take any questions you may have.
Yay, thank you very much. Very well presented. So I'll ask a question while others are thinking of their questions. Lots of applause going on, I see on the screen here. So just raise your hand if you have a question. So my question is, how do your insights and findings differ from previous studies that you've seen?
So previous studies usually focusing on just the effects of one chemical. So usually they have like exposed to BPA in the first generation, and then they examine the effects of that, or PFOS alone. But in our study, we're focusing on the effects of BPA in the first generation. And then after they reproduce, they would be exposing to the newer chemical so that is, if you want to think about it, it's like, let's do, my grandparents were exposed to BPA in their time, right? And then my mom is experiencing to the new chemical of PFOS, which is prevalent in her time. So we want to know if what would happen to me as her children, right? If that's going to give you like a protective effect, or if it's going to be worse, is it going to be better? So we want to look at that, but other studies have not been doing that.
All right, thank you. Who else has questions here for her? All right. Let me go to another question here that I wrote down when I read your paper. What was the most challenging aspect of your research, Kara?
That is a great question. When we are analyzing this data, we see like a U shape, right? Like the control, why is the control having more defects than the BPA lineage? And then when we go up into the concentration, why is it having like BPA lineage has more defects? So that's the hard part in analyzing it, because what we typically seen is like this trend, right? Like as we increases the concentration, the defects would go up, but this is not the case. So it's like a U shape, and we have to figure out what's actually happening. And so my advisor or my supervisor actually told me like, okay, then the endocrine disrupting chemical is usually displaying the non-monotonic shape. So that's the hard part to analyze this.
All right. Anybody else have a question? Thank you for, whoops, we do. Okay. Sorry, Co.
Hey, Kara, that was a fantastic presentation. So I was just wondering if you think that information like this, what you're seeing with the history of exposure from the first generation and then new exposure now, if that's something that you think we can keep tracing into the future generations. So not just maybe the current generation where you're talking about if your mom was pregnant, it's affecting her and that's affecting you. Do you think that maybe if you had children that those effects would carry down as well?
Yeah, so that's a great question. So because this is a transgenerational apple mutations, you carry this in the egg and the sperm cell. And so this can be passed on from generation to generation. And a plus is the environment, or we as the humans are actually creating more chemicals for every generation. So maybe Gen X, which is another more modern chemical, we're experiencing that like every generation. So you would also be seeing this for every single generation and decade. So that would also apply.
Great, thank you.
Okay, thank you very much. Appreciate your time and your being here today. All right, so we'll go to our last presenter for this hour. And Galen, if you want to go ahead and pull up your presentation as I'm reading your bio, that would be great. This is Galen Jones. She's current student at Wayne Community College, pursuing an associate's in arts that should be obtained December of it said 2022. Is that right? Did you just graduate?
I'm sorry. Yes, I did.
Yay, congratulations. Okay.
Thank you.
She is an honors student at Wayne Community College and a member of the national chapter of Phi Theta Kappa. Galen Jones is preparing to transfer from the Wayne Community College in hopes of obtaining her bachelor's, followed by her master's to become a therapist. So you're presenting on a sample scientific research paper on higher percentage of research grade level monarch butterfly observations between the Southwest and Southeastern states. So great. Thank you for coming.
Thank you. Okay, I'm trying to get it to come up and I'm having a little bit of technical difficulties here. Let's see, hold on one second.
We've got Eric if you need help. He is
Yeah,
our tech
well,
guru.
now you've totally went like away. Okay, there you are. Hold on, let's see.
No worries.
Let's try this again. I may need Eric because it is not shooting up from my PowerPoint for some reason.
I'm here. What is
Hey,
it?
Eric.
What is the issue you're facing when you try to share your screen?
It will not connect to my PowerPoint at all, which is really odd.
Interesting.
There we go.
Can you see that?
Yes.
Okay, let's see.
We're only
There's
seeing
a box now.
yeah, we're only seeing a part of your presentation. Do you see like a green box on your screen that's sort of in the middle of your slide?
It's a yellow box
Yellow,
actually.
okay. Maybe if you try, what is it, zooming out on the presentation itself, it should move into the box.
Okay, let's try that.
And the yellow box is showing what we see.
Let's stop that. Let's go back and try to do that all over again. Okay.
I'm going to send you a private message with my email address. In case that doesn't work out, then I can just share the screen for you.
Very
there
good.
you go. Thank you, Eric. Good idea.
Send me your email address. If you send me your presentation, I will happily share it.
Okay.
You'd think after these 3 years that we'd all be experts at doing this, but
You would think. And in my practice, I did this perfectly. And I was like, oh, great. This is going to be easy. And yeah, no.
No worries. It's all good.
Okay, so UNCG. Okay, it should be coming to you.
Thank you,
I
Eric.
am refreshing my inbox until it comes in.
Very good.
Still haven't seen it yet. Do you have a delay on your sending on your Gmail?
It just said message sent.
Oh,
So there
fabulous.
must be a delay.
I have mine set to where emails that I send don't actually send for 30 seconds until afterward, and there's a
Oh,
little
nice.
undo send button. It's a lifesaver.
Are you serious? Oh my goodness. I didn't know it existed.
Oh, wow. Do you see it now?
I do not.
Oh, dear Lord.
It's not my spam either.
Just double check that you got his correct email, maybe. Ah,
Well, his face came up. Can y'all see that now?
Yes.
yes, we
Okay.
can. Yay, thank
Yay.
you. And so now, Eric,
I
you will
had
use the
to
drive.
go like the back route and it craziness. Okay, sorry about that. My name is Galen Jones, and I am presenting on the paper I wrote for my honors biology course last semester, titled A Sample Scientific Research Paper on Higher Percentage of Research Grade Level Monarch Butterfly Observations between Southwestern States and Southeastern States. Before I get into the synopsis of my paper, I wanted to give you a little bit of background on what a monarch butterfly is. So basically, I'm going to give you a couple of characteristics. Monarch butterflies, scientific name is Danius plexippus. They the origin of a monarch butterfly is the American tropics. They can be found in Australia, North America, and New Zealand. They are distinctive for their orange and black wings with the white spots along the trim. And if they are a male, they will have these little black dots on the hind wings. This presents as a defense mechanism to predators because predators automatically think that either they're going to taste bad or that they are toxic. The wingspan of a monarch butterfly is anywhere from 3.5 to 4 inches. And that actually helps out a lot because monarch butterflies are the only species of butterfly that actually does a two-way migration. So they're very similar to birds. The lifespan of a monarch butterfly is very short. It's anywhere from 2 to 6 weeks. And monarch butterflies are natural herbivores that feed off of milkweed and nectar. They sense the milkweed and nectar through their antenna, and they drink through their legs. So for the research part of my paper, I utilized iNaturalist and iNaturalist alone. And for those of you that are not familiar with iNaturalist, it is a superb social network that can be used throughout the world. You basically take pictures or sound bites and upload it to the website, and people can identify the sound or identify the picture and let everyone know exactly like what type of species it is. For my research, I wanted to use research grade level observations. And for that, there has to be 3 specific criteria met. So first, there has to be a date. There has to be coordinates of the observation that is uploaded. And then there has to be a minimum of 2 identifiers that are similar. If there are more than 2 identifiers, then there has to be a 2/3 consensus or agreement on what that species is. So for my question, I came up with, will Southwestern states have a higher percentage of research grade level monarch butterfly observations than the Southeastern states? I had no prior knowledge about the monarch butterflies and how many observations there were. So I did a coin flip and came up with the hypothesis: if data is compared between the Southeastern and Southwestern states on research grade level observations of monarch butterflies, then the Southwestern states will have a higher percentage. So I brought up my iNaturalist webpage, logged in, and from the home page, I selected observations, which gives you this page that you see before you. In the box here, where species is, I typed in monarch butterfly and it came up with the little picture that is directly to the left. I selected that. But because I was going to have a lot of different states to look up, I did not put in a location. I went to the filter section, which brings down the drop box. In the drop box, I knew that I wanted to have a total amount of observations for each state. So I checked verifiable. The category was already selected, so I scrolled over to the dates observed. I wanted the information to be the same for each group of states. So I started with the very first day that an observation was made for monarch butterflies, which happened to be in the early part of March of 2009. The end date I chose as the very first day I started my research, which was November of 2022. So for this, I had to scroll down to where it says place and put in each state so that I would get a total number of observations. So in splitting them up, I had a ton of states to look up. For the Southeastern states, that included Alabama, Arkansas, Florida, Georgia, Mississippi, Kentucky, Louisiana, and North Carolina and South Carolina, and then the territorial areas of Puerto Rico and the Virgin Islands. For the Southwestern states, I included Arizona, New Mexico, California, Colorado, Nevada, Oklahoma, Texas, Utah, Montana, Wyoming, South Dakota, and North Dakota. So again, I had to do this for each state. After I had the total amount of observations for each state, I had to find out how many of those were research grade level. So for my second search, I virtually kept everything the same, but I had to include research grade as part of my filter search. Again, I did this for every single state in the Southeastern and the Southwestern territories. And I was able to come up to make my graphs. This is a graph of the observations in the Southeastern states. As you can see, Florida had the most with a little bit over 7,500 observations. North Carolina, I was proud to say, came in second, and they had a little over 3,800. The territorial islands, the Virgin Islands and Puerto Rico, the territorial areas, had the least amount. Puerto Rico had a little over 100, and Virgin Islands had not even 20. So in the blue, that is the research grade level observations, and in the green, that is the total amount of observations. My next graph is for the Southwestern states. Again, the blue is for the research grade observations and the green are for the total. California and Texas were the two states that had the most observations. And when I said the most, they had a ton. California had over 27,000 observations, and Texas had almost 30,000, 29,000. And then out of those, 26,000 of those, a little more, were research grade levels. Even its states that had the smallest amounts were triple the amount of the Southeastern states. Wyoming and Montana were the least amount of observations. Montana had 51 and Wyoming had 61. So to find out the percentage of research grade observations, I had to calculate all of the total observations for each state, which for the Southeastern state, the total was 43,543 observations. For the research grade level observations, there was 43,454. So they were very neck and neck. For the Southwestern, the total observation was 62,835. Research grade levels were 59,411. So initially, I believed that the Southwestern would be the one to win, but it was not. The Southeastern was the winner. My hypothesis was incorrect. The percentage for Southeastern, because it was so similar, was 99.97%, which is so close to 100%. And then the Southwestern was 94.55. So in the end, quantity was not the determining factor. And that really amazed me because I just knew that with all the observations that the Southwestern had, that they were going to pull it off, but they did not. It was a pleasure presenting for you. This concludes my presentation. I hope you enjoyed it. I will be happy to answer any questions you have, and I thank you all for your time.
All right, thank you very much. Good job. And yes,
Thank
look at the
you.
applause coming through. So I have a question for you.
Yes.
Why monarch butterflies?
Okay, so monarch butterflies specifically are my favorite, but I have always been fascinated by butterflies. It's the intricate detail and the designs of the butterflies. It stems back to childhood.
Okay, thank you.
You're welcome.
Anybody out there have a question for Galen? I can go to my second question. Oh, what is the scientific relevance of your research?
So the scientific relevance, basically, when I was younger, I saw butterflies everywhere, and it might be the same for you. But as I got older and into my adulthood, I have not seen many butterflies. It's like they just, poof, they're gone. And that really troubles me. So in doing the research, it was nice to see that, people are still observing them. They are still seeing them, but they're just not as prominent, I guess, here in North Carolina where I am as they are elsewhere. But in doing the research and finding out that the livelihood or lifespan of a monarch, which is my favorite butterfly, is 2 to 6 weeks, that was really like concerning to me. And I just, I don't know, just always had the soft spot.
Do you know if their numbers have declined over time? Like, is there any way to go and see, like, before, I think you said 2009 was when you started this count?
That was the
Before
first
that date?
observation.
Yeah. Oh, so iNaturalist doesn't go before then?
No, actually, iNaturalist launched in 2008.
All right, anybody else have any questions for her? Okay, well, thank you very much. Good job. This
Thank you for your
concludes
time.
our hour. I appreciate it. And Eric, anything else that we need to do to close out?
Nope, that is all. Thank you all
All
so
right.
much. And I hope you have a wonderful weekend and rest of your Friday.
Yes, for
You as well.
sure. Thank you.
Thank you too.
Y'all.
Bye-bye.
Bye-bye.